So, thanks for the invitation. Today I want to tell you why I'm such a big fan of such a small worm. It's about the nematode C. elegans. And when I say it's small, it's really small. It's just 1 millimeter in length, so you really need a microscope to watch it. So why are so many people interested in such a small animal? So first of all, there are a lot of practical reasons to use it, because the handling is quite easy. The worm lives on small agar plates, and the worm eats E. coli as a food source, which is easy to maintain in any lab. So, yeah. The next reason is that the worm is an hermaphrodite. So the offspring of the nematodes are clones. So you can be sure that your groups which you want to compare, so the treated group and the control group and so on, are genetically the same, which has a lot of advantages. And do you also have a pointer or something like this?
It should be right there next to the laptop.
This one, yeah.
Push it back.
Oh, yeah, yeah. So here you have an example of the worm's body of the hermaphrodite. So you can see he produces sperms and eggs in the same animal, which is quite practical. Moreover, we have a lot of offspring. One worm is producing about 300 eggs in only 4 days. So this is quite fast and quite a lot. And on this picture, you also see some of his larvae. The next reason why we use it, and especially the biogerontologists love to use it, is that the worm has a very short lifespan. So as you can see here, this is a picture from our lab, what we made, that the worm lives at 15 degrees in a median about 22 days, and when you use 25 degrees, it's only about 9 days. Most researchers use 20 degrees, it's about 15 days, the median. And the maximum is usually not more than 30 days then, or here 36. So one lifespan, let's say, is quite fast, especially compared to other animals like mouse, where you need some years, and so one whole PhD student for just one result. The next is that the genome of C. elegans was sequenced as a first animal, completely sequenced. And depending on what you're reading, but some authors suggest that more than 80% of the C. elegans proteome, so the protein sequences, have human homologs. So this is why so many researchers are interested, because they think that the results can also be used for human research later. And what you can see here is a gene chip. This is a microarray. We also heard about this today. This is the consequence that we can, the genome is sequenced, is that we can use microarrays. And just to show you what it is, it's really quite that small. And on this small patch here, all of the genes of C. elegans are represented. So it's more than 22,000 genes are spotted, or not the whole gene, I will show you soon, in several copies. Oh yeah, here. So one gene is in this case, so there are several different microarray types, but this one is what I used usually. We have about 25 base pairs per gene, which is specific for one gene, which is immobilized on this small glass slide, and each gene in several copies. And how a typical microarray experiment looks like, and which I also did a lot of them, so is that you have two groups which you want to compare. There's the normal nematodes and the nematodes which are treated with something, usually something that should prolong the lifespan. The genes will be transcribed to RNA, and maybe the substance can change the rate of transcription, can inhibit or enhance, in this case, it enhanced the transcription, so more RNA will be produced, and later on, probably, also more protein will be produced. But this is also dependent on the translation rate, but you can also have a research on this one because this is a different part, but RNA is quite easier and was first developed. So what you do then is to collect the RNA from both groups and put it on the microarray, and with complementary bounds, the RNA will find their specific partner, the DNA which is spotted on the array. The RNA was labeled before with a special color, so you can see when you scan the color of the microarray, you can see, you can quantify how many RNA was there. I mean, I don't want to tell you in detail that much, but I think the principle is quite easy. So next is that we can, due to the completely sequenced genome, and it was the first one, there are several mutant strains exist from C. elegans. This means that one gene usually, sometimes also more, are destroyed or deleted or somehow changed, that this gene is not functioning anymore. And you can easily order these mutant strains. It's quite cheap, quite fast, so a lot of researchers take this chance. What do we do with these mutants? In my case, when I did these longevity experiments, it's like you have the wild type with no mutant and the mutants, let's say gene B, which have a normal lifespan. Then you test the substance X, whatever, to both of these groups, and when you see that there's a life extension with the mutant strain, with the wild type, and there's no life extension with the mutant strain, you can assume maybe that this gene B, plays maybe an important role in the life extension mechanism of the substance. This is a first hint, which you can go on further. So the last point here is that C. elegans shows typical signs of aging, which is quite important for biogerontologists. As you can see here, this is a day 5 adult, which looks quite healthy. This is a day 16 adult, which looks kind of wrinkled, or yeah, it's quite comparable to humans. But it's not only the looking, not only with the eyes you can see it, you can also measure it in several ways that C. elegans is also aging. First of all, he shows also less and slower movement, and the decreased muscle function, which I guess is the reason for the slower movement. Then you have less stress resistance, an increased danger of infection, decreased protein homeostasis, and cognitive decline. This is something what C. elegans and humans have in common. So we can also make real aging research with C. elegans. Well, in the biogerontology challenges include to prolong the lifespan, but also to retard the aging, which is not always the same. What we find out, and what also other researchers found out, sometimes lifespan is prolonged, but the health span, which is your health, is maybe not improved, or even worse. It's, several cases were reported, so it's important to research both the lifespan and the signs of aging or health span, however you want to call it. And later, sure, you want to transfer the data from the models to the humans, which is maybe the most critical point, because this is not easy. But what did we learn from C. elegans? There are at least 4 proven mechanisms for life extension in C. elegans. The first one is hormesis. And hormesis is a mild stress which has beneficial effects. A mild stress can be something like a toxic substance in such a low concentration, And the beneficial effects could be longer life, enhanced stress resistance, could be also better reproduction, and so on. It is said that hormesis is independent of the endpoint, the organism, and the stressor. Well, there's some, it's not completely clear because there are still some substances who showed no signs of hormesis yet. I mean, there are some controversials about this point, but I think there's more research needed. Maybe they just, they just looked on the wrong endpoint when they had a look on the stressor. I don't know. So in C. elegans, hormesis was shown to work with radiation, nuclear radiation, inorganic and organic substances, and heat stress. And this is a typical hormetic curve. So you have here the response, which could be lifespan or stress resistance and so on. And this is the concentration or the dose or whatever of the substance or the treatment. The possible mechanisms are adaptive stress response. This is the most discussed mechanism, but again, it's still under discussion. It's not clear if this is really true, or if this is the only answer. But the adaptive stress response says that the stress defense system is trained by a small amount of stressor. So when there are bigger stressors come later, especially when the worm is aging or whatever, then the worm is better trained to fight against this stress. And this is how hormesis could work. In our lab, we always used polyphenols to see if they have hormetic effects. And we found some with tannic acid and ellagic acid shown here. So this is the lifespan, this is the concentration. And you can see this small peak here. This is life extension. And later, when you use higher concentrations, it will get toxic, so the lifespan is going down. So the lifespan gets shorter. This is a typical hormetic curve. But we also did some more tests with these concentrations. We used the best concentrations to prolong the lifespan, to also check other life parameters. And then we found that it's not that the connection to health span or anti-aging is not that clear, as I said before. This means that, for instance, the initial reproduction, which means the reproductive starting point, when the reproduction starts, was delayed in 3 or 4 cases of the polyphenols. So the worm starts later, which can mean in nature, which could be critical in nature, just because when the predator comes and eats you and you didn't start to reproduce, maybe your population will die. So it's important to start early and fast. So this is a quite really disadvantage. Moreover, we can see that 2 of the 4 polyphenols reduce the growth, what we measured by the body length. And there was also 1 polyphenol which decreases the oxidative stress resistance. So even if it was the prolonged lifespan, there's a decreased stress resistance. What about hormesis in humans? Well, there are some observational studies mainly, and just some examples. There were some reports about radiation stress, nuclear workers who probably live longer. There was some ischemia stress, so when you have a mild ischemia stress, then you should not die that fast because of a heart attack. Then there's some exercise stress. Sure, I mean, sport is stress for my feeling, but it's also stress from the biological feeling, because when you make sport, the body will produce oxidative stress, reactive oxygen species, and we all know that sport improves the disease resistance, so this is also hormesis. Something like thinking stress, so when you use your brain quite hard to think about a problem, whatever, then the brain will also produce reactive oxygen species, so it's also in stress. But we all know that using the brain is quite good for us and will improve brain function in the long term. The negative effects of hormesis are for instance, the dangerous effects with antibiotics and chemotherapeutics in humans. So when you treat an infection or when you treat a cancer cell with medicine and you don't take enough, then these cancer cells or the bacteria cells could react in a hormetic way, means they even grow better than before. So this is several times proven, and this is quite negative for humans. Moreover, we only have a narrow concentration range, so when you take too much of it, it will get toxic. So it's important for the hormetic effect to stay in a special concentration, which might be completely different for each individual. So this must be determined before taking any toxins. Moreover, there's only really a slight life extension. So it's a good question if it's really necessary to take the risk of this narrow concentration range. Moreover, as I show you that there may be also side effects possible, and the most important but is that longevity is not proven in humans by hormesis. The next one is calorie restriction. So calorie restriction just means that you eat less. So longevity and age deceleration by reducing the amount of food to about 30 to 60% of the ad libitum amount, whereas this 30 to 60% is also under discussion. There are different reports, taking more, taking less. There's so many protocols. In our lab, we did one calorie restriction experiment without food. This means we fed C. elegans only up to the 6th day of adulthood, because we wanted to make sure that he's not that he develops completely and that the reproduction is completed. And after this, we stopped feeding him. And as you can see, there's a huge life prolongation. But we all know that this is not possible in humans. But it was quite interesting to see. Well, calorie restriction works in almost all species. As you can see here, this is some old data. I think we have some new, but just as you have an imagination. So we have the C. elegans, we have the fruit fly, fishes, mice, dogs. These are all animals where calorie restriction were reported, and even more. One theory how this calorie restriction works is that it's a kind of mild stress and acts in a hormetic way as described before. But there are, hundreds of theories. There was a quite interesting experiments with rhesus monkeys, and they're especially interesting because they are close related to humans, so we think about this might say something about if calorie restriction is also working in our body. So they got 30% less food, and indeed, they could see that age-related diseases like cancer, heart disease, and diabetes were decreased, and the brain function was increased. However, the lifespan was controversial, so there was one study found a life extension in the monkeys, and the other one did not. So it's not clear if this is really true. Nevertheless, this poor guy really looks not happy. What about calorie restriction in humans? It's kind of proven that calorie restriction is protective against obesity, what is not really surprising. It's against diabetes, inflammation, hypertension, cardiovascular disease, and cancer. Nevertheless, longevity is not proven in humans yet, so I don't know if we should really take this risk, because the quality of life is kind of questionable. Moreover, the side effects, which are reported in several animals, are reduced growth, development, and reproduction. So maybe this also might not be the right way to prolong our lifespan. The next one what we also heard today a little bit already, is the targeted molecular modulation. Means in C. elegans there are several genes and pathways which are connected to lifespan, to longevity. These are only some examples here. Most of the genes which are connected to lifespan are in the insulin signaling, mitochondrial signaling, germline, neuronal signaling, and food signaling. Here are some examples. So when you inhibit these genes or when you enhance these genes, whatever, you can change the lifespan of C. elegans dramatically. And for one example, there's more than 5-fold life extension just because 2 genes are missing. It's quite easy. And mathematically, this would mean, lifespan in humans up to 500 years would be possible. I guess this is not true. This would be too easy to be true. But the effects which you get in C. elegans are very robust and reproducible. Nevertheless, we have a health span impairment in some of the mutant strains. You can see that the movement is slower, or that they kind of confused animals. They like to leave the plate and whether die, some of suicide worms or whatever. So it's kind of interesting what these mutations really do in the animal, because we don't know actually. Moreover, in humans, sure, we have ethical concerns. We cannot just build a new human with 1 gene missing. And another one, it's not proven in humans, and I really think it's not possible to just delete 1 gene and live 500 years. I think it's not that easy. Another point is that it's only active in the offspring. So when in C. elegans, the germline cells will be changed, so the gene will be deleted there, so the offspring then will live longer. So I myself, I would not have any advantage of this, just my offspring. But this is in brackets because there's a quite new technology. So normally when you want to have these mutant strains, you delete the DNA, so you change some bases or whatever, then you have the mutant strain. But today it's also possible to kind of get rid of the RNA of the special gene. This is called gene silencing by RNA interference, or RNAi, which was also developed in C. elegans, this technique, and the worm got a Nobel Prize for this, or his researcher actually. And with this technique, you can inhibit the RNA which matches this gene, so the protein will not be translated. This is kind of the same function as a mutant strain. Indeed, they are trying to use this technique in humans for several therapeutic reasons, and I think when I'm up to date, it's not possible yet, but I'm not sure completely. I don't think so. Okay, next one is deep freezing. I put it here. I mean, normally it's not, when people talk about life extension and C. elegans, this is not the point what normally comes. This is just for this conference here. Because, sure, deep freezing is, I mean, life extension is quite nice, but normally you wouldn't want to be awake. So during the deep freezing, you cannot recognize that you are living longer. But for this case here, it fits quite well. So deep freezing, normally the most researchers who are working with C. elegans use deep freezing just for practical reasons, because we order a mutant strain and we don't want to use it now, maybe in some weeks, months, or years. So we deep freeze it, and later, maybe 2 years later, we say, oh, I need this mutant, and I thought, and I can use it. For me especially, it was only important that at least 1 worm will survive, because it's a hermaphrodite, so I only need 1 worm. And when this one survived, then I can build up a new population. So I did not give that much on quality for the freezing. I just put some glycerol inside, put it in the freezer, and it was fine. At least 1 worm was always surviving. But I found also a better protocol who are very interested in making a good quality of freezing. And they say that, so their freezing procedure is to cool rapidly to -3 degrees with an ice seeding treatment in order to avoid the spontaneous nucleation, and 5% DMSO as a cryoprotectant. The cooling down to 100 degrees is quite slow at 0.2 degree per minute, and after this the worms will put to liquid nitrogen. The survival rate would be, regarding to this paper, 85% for larvae and also for adults, which was kind of complicated in the beginning. So I never tried, but I have to trust them. And the warming should be quite slow. It should be storable up to several years or decades. I only tried for 10 years about, and it really worked. There were still worms alive, not all, but still some. And some more details in this procedure paper. They compared some ice seeding treatments, and they took a steel needle pre-cooled with liquid nitrogen, which is, the technique is shown here. The other ice seeding treatment was the addition of a special bacteria, with this ice nuclear bacteria, Pseudomonas syringae. And they used a control without ice seeding, and they compared everything. In general, it's also working without ice seeding, but you can see that there's definitely an advantage. And just to add these bacteria seems to be quite easy, so they preferred this procedure. Moreover, they compared some cooling, some cooling rates. And as you can see very clearly that the slower the better, but this is also not very surprising. So the question is deep freezing also possible in humans? Ötzi already tried, he failed. I hope that we will have more success soon, and I think this is also part of our conference. That's it. Thank you.
Thank you very much, Nadine. Are there some questions?
Thank you, that was very interesting. I thought you might like to know, this is not yet published, but will be soon, just Alcor recently sponsored some research that we conducted with a researcher coming from Spain, working with Natasha Vidamore, and we did research with C. elegans, not just cooling them and then rewarming them, but actually training them to do certain tasks, to look for food, basically to learn which way to look for food, and then cryopreserving them, rewarming them, and then testing their task memory. And that actually was a success. After a lot of experiments and looking at different solutions, they actually did reveal that it's actually an imprinting form of memory that was preserved through the cryopreservation process, which I think is quite intriguing.
So you mean they made some cognitive tests, some kind of chemotaxis tests or so on?
Yes, all the details will be in the paper. Basically, they learn which way to go for the food, they follow the concentration of the food, and so they go in one direction rather than another, and that actually was preserved throughout the cryopreservation.
Ah, okay. Sounds promising.